Sharma.
And I'm Ilana
And today we'll be talking to you about Teton region mountain lions, about their overall health and population structure. So mountain lions, pumas, cougars, they're all the same. They have over 40 different common names. So why mountain lions? As far removed as they are, mountain lions are very important to our ecosystem because they're keystone carnivores, which means they help keep prey populations in check. Using viral genetics, we can determine the population structure of the mountain lions in the Teton region, and through that we can determine their overall health. Mountain lions recently have experienced habitat loss and population fragmentation due to human activity and expansion, which limits their genetic variability and decreases their overall health.
So what is FIV? FIV is feline immunodeficiency virus, which is similar to HIV but for cats. Just like HIV, FIV lowers the immune system, making the individuals more susceptible to viruses such as the common co-infections listed below. It is a type of virus which incorporates its genetic material into the host genome, which allows us to detect it using genetic analysis. It mutates at a faster rate than host DNA, so it gives us a snapshot picture of what the population structure looks like currently, as opposed to the host DNA, which provides a more dated look. So what did we do? DNA was extracted from blood samples from mountain lions in the Teton region of Wyoming. To detect the pathogens, we used PCR, which is polymerase chain reaction, which is pretty much like a photocopier that makes billions of copies of the specific region that you're looking for, and it comes out in a nice little tube. And the products were then analyzed and gel purified. It was then sequenced. We tried multiple times to sequence, and we came up with that our sequences were not of high quality. So our goal was to analyze sequence variability to group the mountain lions into subgroups based on their mutations. We were unable to do this, but we were still able to assess their health by looking at other pathogens.
So while we didn't use sequences, we were able to use the gel to test to see if our individuals might have these pathogens. So this is a gel of our 47 individuals, and 35 of the 47 individuals, which is approximately 75%, showed positive amplification for FIV. So that means anywhere you see these white bands, that individual mountain lion most likely has FIV, but we could not confirm it 100% without the sequence. So then we also tested our individuals for T. gondii, which is another co-infection, and 30 out of our 47 individuals, which is approximately 65%, showed positive amplification for T. gondii. And this is a gel showing our 10 most recent samples that we added to the study. These two down here are positive controls. These are 10 samples, and any sample showing a band in this region means that they most likely have T. gondii.
Our results showed that the population as a whole had the highest infection of FIV and the lowest infection of Yersinia pestis, the plague, and no infection of Bartonella. We also found something interesting, that females, the red bar, seem to have a higher infection of Toxoplasma as well as a higher co-infection of FIV and Toxoplasma. This could possibly be because females tend to eat smaller prey, which are usually carriers for Toxoplasma.
So what does this indicate about the overall health? Basically that while FIV and T. gondii are not particularly harmful to our mountain lions, a lot of them are infected with the virus and these infections, which suppresses their immune system, which means that in the future that these mountain lions are more susceptible to getting other viruses that are very harmful to them, such as the plague. Basically saying that we need to watch out for these mountain lions. So thank you, AMNH, the Teton Cougar Project, for the samples, NSF, our mentor, Anthony.
And our mountain lions.
Thank you.